Hello everyone, myself Ms.Namita Garude, Assistant Professor in Microbiology. Let us study Haemocytometer method. Haemocytometer is a special laser-edge glass microscope slide used to determine the concentration of the cell. The device used for the determining the number of cells per unit volume of a suspension is called a counting chamber. When the haemocytometer was invented, used for performing blood cell count only, but nowadays it is used to count other type of cells and other microscopic particles as well. Haemocytometer was discovered by the Louis Charles Melazes. It is a special type of microscope slide which is heavy in a weight, consists of two chambers. One is a red blood cell chamber and other one is white blood cell chamber. It is divided into 9 large square of 1 by 1 millimeter, which are separated from one by another by triple lines. Cover glass is supported over the chamber at a height of 0.1 millimeter. RBC chamber has a 25 square and these are small in size. WBC chambers has 16 squares, large in size. Viability of the cells can be determined by using different stains. Dyes used such as trypan blue, eosine or propydium. Live cells contain intact cell membrane that eliminate the certain types of dyes and appears colorless. Whereas in dead cells, the stain enters the cytoplasm and the cell take up the stain and appeared color. If more than 25% of the cells are stained, the cell suspension is most likely not a viable one. This is the structure of one side chamber of the haemocytometer. It contains 4 different WBC chambers and 5 RBC chambers. The requirements are cell suspension, haemocytometer slide, micropipette, 70% alcohol, compound microscope, dye, trypan blue, clean cover slip. Procedure: first take clean and dry glass slide, haemocytometer slide, wipe it with the 75% ethanol to disinfect the haemocytometer slide, put a cover slip onto the slide, add the suspension of bacterial cell with the trypan blue and observe under the microscope. This is the diagrammatically representation of the procedure of how to use haemocytometer. How the bacterial count is done by using haemocytometer in this photographic presentation. Calculation: the number of cells per ml is equal to n into dilution factor into 1000 divided by area counted, that is number of squares into depth of the field, whereas n is the number of cells counted. Percent cell viability: total number of viable cells which are unstained divided by total number of cell which contains viable as well as dead, means colored and non-colored cells, into 100. Thank you.